Thanks for tuning in. I'm Evan Palmer-Young, presenting from the U.S. Department of Agriculture's Bee Research Lab. This video explains our recent article, Honey, You Light My Fire, Host-Driven Temperature Dependence of Viral Infection in Honeybees, in which we compare the effects of temperature on the physiology of deformed wing virus, honeybees, and virus infection. You can find it on bioRxiv at the URL in the description. Temperature is a fundamental driver of enzymatic and biological processes. One process that can be profoundly influenced by temperature is infection of hosts by parasites. A common response to infection in humans and other animals is fever, an elevation of body temperature. These high temperatures can directly inhibit parasite growth, potentiate host immunity, and ameliorate infections in mammals, reptiles, insects, and even plants. The effects of temperature on infection reflect changes in the relative performance of parasites and of hosts. All else being equal, we expect that a strong parasite will be more capable of infection, and a strong host will be better able to resist this infection. Hence, the outcome of infection will reflect a balance between performance of parasites and hosts. So temperature can affect this balance and, as a result, change the outcome of the infection. For example, parasites may be better able to infect a cold-adapted host at warm temperatures, but infect warm-adapted hosts better at cold temperatures. We can summarize this with the Art of War quote. In war, the way is to avoid what is strong and strike at what is weak. Honeybees provide an intriguing system in which to examine how temperature-dependent changes in the relative performance of parasites and hosts affects the infection outcome. The thermal biology of honeybees makes them unique among the potential hosts of insect parasites and may challenge the establishment of infection in the colony. Honeybee colonies exhibit an endothermic temperature regulation that's quite rare among insects, keeping the colony at close to mammalian body temperatures. This is much hotter than what's typical of most insects, which are generally cold-blooded and could deter establishment of parasites that are weakened by such high temperatures. In addition, honeybee physiology is adapted to perform well at these high temperatures, which are necessary for both successful development and flight. Hence, temperature elevation could not only weaken parasites, but also strengthen the ability of bees to resist them. Indeed, several types of honeybee infection are reduced by high but still colony-realistic temperatures. In this study, we focused on honeybee infection with deformed wing virus. This is considered the most prevalent virus in honeybee colonies, found in over half of colonies around the world, and high infection levels compromise bee development and survival, sometimes quite severely, as indicated by these deformed wings. With its spread assisted and virulence amplified by the now ubiquitous varroa mite, DWV has emerged as a likely driver of colony collapse. Many high-intensity infections are acquired during feeding of mites on developing pupae at the well-heated core of the colony. But despite its success in honeybees, DWV is likely a generalist parasite, having been detected in 8 orders of generally ectothermic arthropod hosts. And it's likely not so preferentially adapted to high temperatures as our honeybees. But the effects of temperature on pupal infection have not been studied, nor the relationship of such infection to the relative performance of parasite and host. To examine how the temperature dependence of infection is shaped by changes in the performance of parasites, DWV, and hosts, the honeybee, we compared effects of temperature on the activity of an isolated virus enzyme, 3 honeybee traits using previous authors' data, thank you, and infection of virus-injected pupae. Use of this isolated virus enzyme allowed us to work around the inability to grow viruses outside of host cells, allowing us to better separate the likely effects of temperature on the virus itself from its interaction with host cells. We predicted that the virus enzyme would maintain activity across a wide temperature range, consistent with its distribution in a wide range of ectothermic hosts, whereas honeybee physiology would be highly temperature sensitive, achieving peak function only within a narrow band of high temperatures. As a result, parasites would be at a progressive disadvantage with increasing temperature, resulting in reduction in infection over the temperature range found in colonies. One more time, now with illustrations. We predicted that the virus enzyme would maintain its activity across a wide temperature range that characterizes its mostly ectothermic hosts, as suggested by an initial characterization at 4 temperatures that showed only around 20% variation in rate across a 25-degree temperature interval. Whereas honeybee physiology would be highly temperature sensitive, achieving its peak function only within a narrow high temperature band, as exemplified by these data on muscle force production. As a result, parasites would be at a progressive disadvantage with increasing temperature as function of the virus remains stable or begins to decline, while performance of the honeybee rapidly increases, resulting in a predicted reduction in infection over the temperature range found in colonies. Now, how about our results? For the isolated virus enzyme, we did find a remarkable conservation of function across temperatures, with activity maintained within 50% of its maximum value over a broad, more than 30-degree temperature range that spanned the typical operating temperatures found in ectothermic insects. The 3 bee traits, on the other hand, were strongly responsive to temperature and relatively high temperature specialized. For example, larval respiration, a measure of whole body metabolism, increased 8-fold over a 20-degree range up to 38 Celsius. And pupal development was even more temperature sensitive, with development only completing across a narrow 8-degree temperature band. Peak activity for all the traits was projected to fall at or above 35 degrees, reflecting the upper end of the temperature range found in colonies, and the muscle temperatures that bees maintain during flight. This well-defined peak of bee performance at high temperatures was in striking contrast to the virus enzyme's broad peak of activity across a range of relatively moderate temperatures. Again, suggesting that performance of the virus would decline both absolutely and relatively to its host over the typical range in the colony, between 20 and 37 degrees or so. However, the temperature dependence of virus infection in bee pupae was almost identical to that of pupal development, increasing by around 6-fold up to 34 degrees, then falling sharply by 98% at pupae's upper thermal limit of 37 degrees. In other words, the success of the virus in bees appears to correlate directly with the performance of the host. rather than inversely as predicted based on the relative performance of parasites and hosts. Note that this trajectory is also quite different from that of the isolated virus enzyme, suggesting that infection is not driven by temperature-dependent changes in physiology of the virus itself. So this raises the question, why should a strong host support rather than quench the proliferation of its parasite? And one explanation may lie in the host-dependent nature of virus proliferation, which requires a functional host cell for translation of the viral genome and production of new viral particles. A strong host metabolism may make new viruses at a faster rate. And although this explanation seems intuitive for a viral parasite, it nevertheless contrasts with several other arthropod host intracellular parasite systems, in which infection progresses most rapidly outside the host range of peak function. Our results also suggest the limited efficacy of the bee immune system against viruses that are introduced by injection during the pupal stage, consistent with the vigorous proliferation of viruses that are introduced directly into the body cavity by varroa mites. This contrasts with the oral exposure route that's believed to have predominated in the pre-varroa mite past. Now, could a colony fever be an effective self-medication strategy for honeybees? Based on our data, although high temperatures can strongly reduce infection and within the range achievable by bees, replication of the virus is closely matched with performance of pupae. Hence, the high temperature mediated reductions in virus levels seem likely to endanger the health of developing bees. This is consistent with a previous study of DWV infection in adults, where 37-degree high temperatures reduced virus infection, but were also very costly for host survival. That being said, honeybees can regulate colony brood temperatures within a very narrow range. We can't rule out that there is a narrow temperature window that would reduce virus levels without compromising bee development, but it doesn't seem all that likely, given that this key virus enzyme retains some activity at temperatures that are beyond the thermal limits of pupae, indicating that the virus can tolerate higher temperatures than its host can. So reflecting back on our potential explanations for infection success, temperature dependence of infection was not well predicted by the activity of the isolated virus enzyme up here, or by the relative performance of viruses and hosts. Instead, virus proliferation rate was directly linked to host performance, indicating a primarily supportive rather than antagonistic role of the host physiology in infection. This result indicates the importance of considering the parasite facilitating and not just the parasite limiting function of hosts. So we could summarize DWV infection with another art of war quote: "The expert in battle moves the enemy and is not moved by him." For DWV, conservation of enzyme function across a wide temperature range explains its presence in a diversity of insect hosts, while in honeybees, its ability to exploit host physiology once inside the body cavity, proliferating fastest in the preferred temperature range of the brood-rearing colony, explains its success as a global epidemic of managed honeybees. And whereas a bee fever might be a plausible strategy for reduction of infection, such a temperature elevation might weaken the host more than the virus itself, rendering it a questionable option for self-medication. It will be interesting to see how the temperature dependence of infection varies across different routes of infection, which might vary in exposure to the host immune barriers, and across hosts with different temperature sensitivities and optima. To sum up a poem, though high temperatures needed by bee, plants and insects of viruses free, instead of parasite toast, seems warm blood of bee host stokes the fire of DWV. With that, I'd like to thank my sponsors and colleagues, the authors of the studies from which the bee trait models were compiled. Those are cited on the next slide, and you for your audience. If you want more, please check out our preprint on bioRxiv.